we did not finish the electrolyte balance. We have 3 more to cover, I believe. They're pretty straightforward, and I will explain, okay? So we're gonna continue with a couple of 3 more electrolytes. Calcium, okay? Calcium is extremely important. We have mentioned calcium for muscle contraction, nerve function, so that is really the centerpiece for the calcium center. We talked about it with respect to the heart too, didn't we? So calcium levels must be kept within a narrow range. If you ever get a blood test, they are going to test your calcium levels and find out if it's in normal range. They always tell you the range and tell you if you're in there or not. Calcium concentration in the blood, calcium levels are so important, your body is willing to sacrifice bone integrity to keep your muscle and your neurons working properly. So that's really, really important. When calcium levels drop, hypocalcemia, the parathyroid hormone will be secreted. When it's secreted, it does the following. Your calcium levels are too low. To bring them up, the first thing it does, osteoclasts become more active and start breaking down more bone matrix than it was before. And of course, when it solubilizes the bone matrix, it puts the calcium and the phosphate into the bloodstream. Make sure you pick up a copy of what we're covering. And make sure you take a picture, a copy of this. Okay? So you're willing to have osteoclasts go start tearing down bone matrix to solubilize both the calcium and the phosphate in the bloodstream so that it raises calcium levels, okay? The parathyroid hormone will also stimulate the renal calcium absorption. This is in the distal convoluted tubules. Everything that happens in the proximal is obligatory. And so calcium will be reabsorbed to a higher level than it was before. More parathyroid hormone, pull in more calcium. And notice the phosphate is secreted. Now, when the osteoclasts degrade bone matrix, it releases hydroxide ions, it releases phosphate ions, and it releases calcium ions. We're now getting rid of, we're actively secreting phosphate ions in there. Why are we secreting phosphate ions? First of all, you don't need them. That's a separate ion that's kept level. But what other reason? We're not affecting osteoblasts, are we here? Do we want to give osteoblasts more material to work with to build bone matrix? Remember, we are stimulating osteoclasts to break down bone matrix so you get calcium. You don't want your osteoblasts to have too much to work with as far as calcium and phosphate. So we're going to start secreting phosphate, reducing that a little so the osteoblasts aren't as active as they were before. Now they're still going to be active, just not quite as active. So that's the reason the phosphate is secreted. Okay? Parathyroid hormone is also going to increase the activation or the formation of vitamin D3, which is the active form of vitamin D. And having more vitamin D is going to increase the calcium absorption in your intestine for that iron. So when your calcium levels go low, you're pulling calcium ions out of the bone, you're reabsorbing more in the kidney tubule, and you're pulling in more from your gut contents. All 3 are necessary. For example, if you're not getting enough vitamin D, then you're not going to absorb hardly anything. And yes, you'll be pulling them out of bones, but you're going to be not pulling as much. You're going to be pulling a lot out of your bones to bring it back up if you don't have enough vitamin D. So if you buy calcium in the store, calcium supplements, you'll read it always contains vitamin D. To make sure you have enough vitamin D to absorb the calcium you are taking in pill form. Ever wonder why it is? You can't get straight calcium without vitamin D. It always has vitamin D because of the need of having having adequate amounts to absorb the calcium in your gut. Okay? If a female becomes postmenopausal, she does not have adequate levels. Make sure you pick up a copy of today's notes back there in that corner is the only location. If a woman is postmenopausal, she has no estrogen, and estrogen encourages the putting of calcium into bones. In that case, they can be taking it and reabsorbing, but osteoblasts are not as active as before, so they're not actually putting it in the bones. And they can end up with osteoporosis, as every time it drops, you pull it out of bones, and when it rises back up to normal, you don't rebuild it. So that's a problem. So you need to make sure calcium levels in a female stay high, so they ever will stimulate these osteoblasts. They pull it out, and then you don't have enough estrogen to cause the osteoblast to put it back in when it gets back up to normal. Okay, whenever calcium levels increase, and this is rare in an adult, when they go too high above out of the normal range, parathyroid secretion declines, so you're gonna have less osteoblast activity, you'll have less calcium reabsorption in the tubules, you'll have less calcium absorption in the gut, in the intestine, okay? And the calcitonin is going to be secreted, and that inhibits osteoclasts. So unlike other ions where it's not very robust control, this is extremely robust, powerful control. If it ever goes too high, it's going to come up, even if your bones get weak. And whenever it's too high, you're going to get rid of the calcium, because osteoclasts will be inhibited, so they're not allowed to break down any bone. Osteoblasts merely keep doing what they're doing, building bone and lowering it this way. When PTH secretion declines, remember, you have less vitamin D, less calcium reabsorption in the renal tubule, and the osteoclasts are already going to be inhibited instead of stimulated. So understand, this is a very robust, tight control. There's no other ion that has this level of control. Shows you how important calcium is for our bodies. Magnesium. Okay, magnesium is, this control is just happenstance with the kidney. Okay? Magnesium ions are extremely important to have because they're cofactors for some enzymes, especially those involving ATP and that kind of thing. So they're very important enzymes. And you would think the control is pretty robust? Not really. If magnesium levels are too high, they go into the filtrate, they recover some, and the excess is going to be secreted. Well, excreted in the urine. So if it's too high, you just don't absorb the excess and it goes into the urine. If it's too low, virtually all the ions that go into the filtrate are recovered. And assuming you're taking in adequate food, it'll come back up. If you're taking in adequate food and you have some magnesium ions, that's how it's controlled. That's not near as tight as calcium, is it? That's kind of happenstance with the regular amount of absorption in the proximal convoluted tubule. It absorbs a certain amount. If there's too much going to the filtrate, it's excreted. Levels will drop. If there's too low a level, virtually all the calcium is going to be restored, and you can, if you're assuming you're eating magnesium ions, it'll go up. The same kind of control is there for phosphate ions. If the levels of phosphate ions are too high, the excess isn't reabsorbed. If it's too low, virtually all the ions, phosphate ions in the filtrate are going to be reabsorbed. Okay? The control of those two is very, very simple. Does everybody understand that kind of control? Okay. It's just based on the ability to absorb a certain quantity from the filter. Now, let's go to the last topic, and this is a long one. We're going to look at acid-base balance in the human. In the human, our body fluids, and I'm thinking extracellular fluid or just simply the plasma, has to be kept around 7.4. That's 7.4 plus or minus 0.05, 5/100ths of a pH unit. So normal is 7.35 to 7.45. It's got to be kept that close, extremely tight. When it gets out of that range, proteins don't fold right, enzymes don't work right. It is a serious disorder when it gets out of that range. We have names for just going to 7.46 or 7.34. Okay? It is serious when it gets out of normal range. A lot of things go wrong, not just a couple. A lot of things can go wrong, okay? During normal cellular metabolism, we generate acids, and those extra hydrogen ions must be eliminated every day. It's a large number. I had sound effects there planned for that. Okay, does everybody understand? We generate a lot of hydrogen ions. And those hydrogen ions every day must be eliminated from your body. What do you think is the major way of getting rid of those? Kidney is the major one for getting rid of those. That's a large amount of hydrogen ions. Okay? So that's where they're mostly going to be eliminated. Okay? Everybody understand that point? Now let's talk about some basics about, oh wait, yes, let's do it. Let's talk about some basics on acid base. I know you may have heard it, but how many of you feel uncomfortable talking about acids and bases and pH? Nobody willing to admit it? Okay, here we go. Now, acids and bases are, if they're a strong acid, and you put it in solution, hydrochloric acid, you have none of these molecules floating around. You have only hydrogen ions and only chloride ions. It completely dissociates. That's a strong acid. If you use a strong base like sodium hydroxide, you can put 1 billion molecules into solution. It's a solid. You can put it in solution, you end up with 0 of those left. You have 1 billion of those and 1 billion of those. Completely dissociates. That's what strong acids and strong bases do. Do not get involved personally with those strong acids and strong bases, okay? Now, a base is defined as the ability to produce hydroxide ions or absorb hydrogen ions. Strong acid generates those hydrogen ions. And they always write acids which way? Hydrogen first. What's sulfuric acid? H2SO4. Phosphoric acid, HBO4. Any molecule that starts with an H will release a hydrogen ion. What's the formula for water again? H2O. Does water release a hydrogen ion? Yes, it does. It's a weak acid, but it does release hydrogen ions. Okay, enough of that. Now, we're not concerned about the strong ones. We're concerned about weak acids and weak bases. If you take a weak acid, like, what's this one again? Carbonic acid, okay? H2CO3 is carbonic acid. When you put it into solution, it only partially dissociates. See this? Instead of a one-way arrow, we have an equilibrium. After you put carbonic acid into water, you end up with these molecules, those, and those. Now, I don't know the equilibrium constant for it, but let's say you put 1,000 of these into solution. You'll end up with 500, 500, and 500. You have some of everything in solution. Does everybody understand? And weak acid partially dissociates and releases some hydrogen ions. And this is a weak base, by the way. That bicarbonate ion is a weak base because it can join with and take an excess of hydrogen ions. Weak acids release a hydrogen ion, and a weak base can bind a hydrogen ion. The weak acids and weak bases are important in all physiology. So we're going to start talking about, there's an equilibrium between them. They partially dissociate. So let's talk about how our physiology is set up to regulate acid-base balance, or the pH of our fluids. We have 3 homeostatic mechanisms that regulate the pH of our body fluids. The first is the presence of buffers. We'll explain what a buffer is in a minute. We'll take them first. Then we use the respiratory system to adjust our fluid pH. Yes, we do. And you already know that, but you didn't connect the dots yet. I'll connect them for you. And then finally, of course, the kidneys are able to deal with acid-base pH. So these 3 together maintain, these 3 mechanisms maintain the proper pH of our body fluids. So let's take the buffers first. Buffers prevent extreme changes to pH. If I was to take 2 containers of water, one has a buffer, the other doesn't. If I add a bunch of hydrochloric acid to water, the pH is going to drop drastically. Add the same amount of pH to that buffered solution, it drops maybe a little bit. They prevent those extreme changes to pH when you add hydrogen ions, or even if you remove hydrogen ions by adding a strong base to it, like sodium hydroxide. Buffers are chemicals in solution that prevent drastic changes to pH. I don't know if you've ever, do they do chemistry experiments where they show you how buffers actually work? Yeah. I mean, you add it to water, the pH goes boom. Add it to the buffer solution, it drops. It's hard to measure sometimes the change, isn't it? Yes, they're significant. We have those in our blood to prevent that pH change. We have buffers in our blood, and we're going to look at the 3 kinds of buffers. Buffers consist of a weak acid and a weak base in equilibrium. Did we see weak acid and weak base in equilibrium on the previous slide? Okay, the one before that. Yeah, right here. This is a weak acid, and that's a weak base. And they're in equilibrium. So if we had those chemicals in our blood, we would have a buffer. Actually, it is there. When did we see this carbonic acid? When did we see that? Pardon? In the red blood cells for carrying and also for respiration. So it was in the red blood cells and it's respiration. So we've already seen that. Yes, that is one of the 3 buffer systems in there. So buffers quickly stabilize the pH because they can release or bind hydrogen ions depending on what's needed. Now, buffers work great at preventing pH changes, but they have a limited or finite capacity. Now, I don't know, I'm just going to make up an experiment. Let's say we had water in a buffered solution, and we're adding 2 drops of hydrochloric acid, 2 drops of hydrochloric acid. This one drops a lot, this one drops a little. Now, you can keep adding hydrochloric acid. Eventually, you're going to titrate out and use all the buffering capacity, and then it drops. So they have a limited capacity. Okay? Buffers act quickly. They can release hydrogen ions if needed, absorb hydrogen ions as needed. But they can only do that so much. You have to keep in mind, it's a limited capacity. Okay, let's look at the 3 buffer systems in our body. The first one is the one we just looked at, the carbonic acid bicarbonate buffer system. This is a weak acid, partially dissociated. This is a weak base. Now, that is formed by, of course, carbon dioxide and water. And we have a lot of this in our bloodstream. Why? Because we have dissolved carbon dioxide in our bloodstream, right? So in the bloodstream, we have carbon dioxide dissolved, so we have this and this in equilibrium. If we were to somehow magically remove all the carbon dioxide out of your bloodstream, you'd have very little of that. But we don't ever have that. Okay. This is an important buffer in the extracellular fluid. That would be, again, what are the two compartments, subcompartments of the ECF? What are the two large ones? Plasma, interstitial fluid. So in those two compartments, we have a lot of this in solution, and it's the major buffer in those systems. Not the only buffer, but the major one. Okay? This buffer system only works if the respiratory system is functioning. Okay? It has to be functioning normally, or you don't have all of this. Let's say your respiratory system is compromised and CO2 builds up. We're going to have abnormal levels of those. And this buffer system will not work if pH changes are caused by an excess or a shortage of CO2. If you have too much CO2, too little CO2, this buffer system can't work because you're putting and removing CO2. So this buffer system only works if your respiratory system is functioning normally and if you don't have an excess or shortage of CO2 in your bloodstream, the fluids. Okay, let's see how it works. Okay, we'll explain. Let's say we now have generated, our respiratory system is working normally, okay? We have normal levels of this, normal levels of CO2. If I suddenly do some anaerobic respiration and dump lactic acid into my bloodstream, what's going to happen? I'm going to add hydrogen ions. They're going to build up. Let's say I have 500 of these, 500 those, and 500 those. And if I add 100 more of these, most of them are going to bind with the bicarbonate and push that equilibrium this direction, forming more carbonic acid molecules. They have to be kept in the right ratio. Now, it may absorb 90 of them, so we would have only increased a little. It'll absorb 90, only increase 10, so the pH dropped just a small amount from that lactic acid. That's how it buffers it. We generate acids every day. They have to go into our bloodstream to be eliminated. In the bloodstream, those hydrogen ions push this equation this way, and the pH of the blood doesn't change very much. Does everybody see how that worked? I added 100 of these. I mean, lactate molecules over 2, but we're ignoring them. 100 of these, 90 of them joined, I only increased 10. It's a small change. Everybody understand how that buffer works? Okay. So let's go on to the second buffer. This is the protein buffer system. Now you've heard of this one before too. Now, proteins contain a large number of carboxyl groups attached to amino acids in at one end, and a bunch of amino groups to amino acids, a part of amino acid structure, and at the amino end of a protein chain. So this carboxyl group is a weak acid. If you remove some hydrogen ions, this hydrogen ion will leave the carboxyl group. If you add hydrogen ions, that hydrogen ion will bind to this and form an NH4+, NH3+. Got it? So proteins are, where there's proteins, are there excellent buffer, just the proteins themselves. So this is important in the ICF and the plasma. Why isn't it important? That's inside of cells and in plasma. Why isn't the protein buffer system important in interstitial fluid? They don't have protein. Plasma contains a lot of protein. Inside of cells, lots of protein. And so in those two compartments, there is abundant buffering from the proteins. They always are in those compartments. Everybody clear on that? Okay. Where did we see a protein buffer system before?
Is that intercellular or interstitial?
This is, no, not interstitial fluid, this is intracellular fluid. This is inside of cells. Inside of cells and in plasma. Interstitial fluid, no protein. Okay? So the ICF is intracellular fluid. We had that abbreviation previously in the first part of this when we're talking about fluids. Okay? Make sure everybody knows that's inside of cells. If you're not sure of it, write it next to it so you keep it straight. Okay, so where have we heard of this protein buffer system before? Not transamination, not deamination, neither of those. The hemoglobin absorbing the hydrogen ions when we had this reaction going on to form hydrogen ions. It was we removed these by shipping them out of the cell. Remove these by binding to amino groups in the hemoglobin. There's lots of hemoglobin in there. Now you remember it? It was a protein buffer as it absorbed the hydrogen ions, the amino groups of the hemoglobin. That allowed the red blood cell to produce more bicarbonate ion and allows the blood to carry more carbon dioxide. You remember it now? Good. The other one's a phosphate buffer system. The phosphate buffer is found wherever you have large amounts of phosphate ions. So this is dihydrogen phosphate with a single negative charge, and this is monohydrogen phosphate with -2. This is obviously the weak base, and that's the weak acid. Everybody clear on that? Now, this is an important buffer, again, inside of cells where there's lots of phosphate and in the urine. It doesn't mention urine, but it is in the urine, and they actually show it later on when we talk about how in the renal tubule, how it carries extra hydrogen ions. It shows the phosphate buffer system right there. Now, the book doesn't say it's in the urine, but make sure you do see that. It just says it's important inside cells. It's also very important inside the urine to allow it to carry more hydrogen ions. That pump, bringing sodium in and pumping hydrogen ions out, can only generate at such a low pH. You have to be able to buffer it and carry the hydrogen ions with other molecules to carry sufficient hydrogen ions. If this was not in the buffer, we couldn't get rid of many acids. It's an important one there. Okay, so 3 buffer systems: carbonic acid bicarbonate, proteins, and the phosphate buffer system. Okay, they're important, each one. What's the most important one, do you think, in the interstitial fluid? The carbonic acid bicarbonate. Make sure you understand these, know the components, the molecules, and everything. Okay? So the first change that happens when ions are added or removed, hydrogen ions are added or removed, is the buffers respond first. Remember, they're extremely quick, but they have a limited capacity, and the hydrogen ions are not removed from your body. They're not. Okay? They're still inside your body. Respiratory regulation. Your respiratory system can also help regulate the acid base or the pH of body solutions. It's going to use a carbonic acid bicarbonate buffer system. And again, CO2 plus water, carbonic acid bicarbonate ion, hydrogen ion. If you ever increase the carbon dioxide concentration of any solution, you're going to lower the pH because pushing the equation this way. You add more CO2, pushes it this way, pH drops. It always happens, by definition. You don't need carbonic anhydrase there. It just makes the reaction go quicker. Whenever CO2 builds up, you're gonna lower the pH. Okay? If you remove some CO2, what's gonna happen to the pH? It's going to go up, whatever it was before. You start removing CO2, you're gonna take some more of these, form this, and it'll dissociate, because they're both in equilibrium. Remove CO2, pH goes up. Add CO2, pH goes down. And that's exactly how your body regulates pH, using the respiratory system. When the pH of the blood drops for any reason, when the pH goes down, your rate and depth of breathing will increase. We already mentioned that. When CO2 builds up, pH goes down, and your breathing increases. The rate and the depth of breathing will increase, reducing CO2. Remove CO2, pH goes down because you're removing these from the solution. It's a very simple relationship. So in essence, our breathing rate is controlled by the pH of the extracellular fluid. pH goes down, breathe faster. pH goes up, breathe slower. So if we have done anaerobic respiration, we're gonna be dumping a lot of lactic acid into our bloodstream. So we're breathing faster because we gotta get rid of CO2, but you're breathing faster because all that lactic acid is lowering blood pH. They work together. It's a good thing they don't cancel each other. Okay? So if you have generated a lot of acid into your bloodstream, how do you get rid of it? Respiration. You're gonna take CO2 you're gonna take a bunch of bicarbonate ions, take some of those hydrogen ions from that acid and form a CO2 and remove it from your body. When you take a hydrogen ion from lactic acid, put it with a bicarbonate ion, and we remove the CO2 of that respiratory system, where now is that hydrogen ion? Where'd it go? It's locked up in a water molecule. So you are removing the acid from your body when you use your respiratory system for any acid, give the hydrogen ion, you lock it in a water molecule when you remove a CO2. Does everybody see what happened? You have removed it from your body. Does a buffer remove it from your body, except for this one? No. Buffer still has the hydrogen ion, you have less weak base. This one uses a weak base, locks it up in a water molecule, and you remove a CO2. So this is one method to remove the hydrogen ion, the acid, from your body. The acid, no matter what the name is of the acid, it always releases this, and you can remove it from your body by removing a CO2 from your bloodstream. Doesn't matter what that acid is, you have locked up that hydrogen ion in a water molecule.
Basically, it just gets excreted.
It is in a water molecule. It's not excreted from our body because the water molecule is still there, but you have locked it up in a water molecule. It's as though it doesn't exist in your body anymore. The hydrogen ion is gone from your body. It's no longer free hydrogen ion. Removing a CO2 from the extracellular fluid will tie up a hydrogen ion in a water molecule. Key thing, we eliminate acids by removing a CO2 from the solution that contains the acid. So whenever we generate a lactic acid, it'll get dumped most oftentimes in the bloodstream. We remove it with our respiratory system. Respiratory system begins quickly. Not as quick acting as a buffer. That's virtually instantaneous. Okay? But it begins quickly in a few seconds, and it has a limited capacity. Why does it have a limited capacity? Why is this limited? You only have so much bicarbonate to absorb it and to be able to make a carbon dioxide in the water. Once you run out of this, you're out of the weak base, you cannot absorb a hydrogen ion. It will stay in your blood and the pH will drop. Got that? Now, you can keep breathing, but you're not gonna be lowering raising the pH because you can't get rid of it anymore after you run out of that. So, like buffers, your respiratory system has a limited capacity. Once you run out of bicarbonate ions that are in your blood, it's over. You can't remove any more hydrogen ions. The pH will then change if you add any more acid to it. Does everybody understand how it's limited? All of the buffers are limited. This respiratory system just happens to use a buffer, and it's limited just like the buffer. I mean, you go to any of the buffers that has a limited capacity to absorb or release hydrogen ions. Everybody clear on that? What's left? How does your, and you know this already, we'd actually looked at hydrogen ions being secreted. Remember, you generate acids throughout the day, you need to get rid of those hydrogen ions we talked about and shown once already how they're secreted. Tubule cells can secrete hydrogen ions. That's both the proximal convoluted tubule and the distal convoluted tubule. Both can secrete hydrogen ions. And when they secrete the hydrogen ions, what happens to the ECF, the interstitial fluid of the plasma? pH is going to go up. Every time it secretes one, puts it in the tubular fluid, the pH of the extracellular fluids, plasma, and interstitial fluid goes up. Okay? So we generate a lot of hydrogen ions. This is how you actively get rid of them. Now, both the kidney and the respiratory system can effectively remove them from your body. Buffers don't, but both can. Okay? And you're going to find out that when they put a hydrogen ion into the bloodstream, they make a bicarbonate and put it into the bloodstream. Okay? And that, again, restores the buffers. That's where we get the bicarbonate ions. The bicarbonate ions that are in your bloodstream are generated by the tubule cells. And we'll see how they do it right here. So, this is just an overview of secretion of hydrogen ions. Again, you're going to you can have a hydrogen ion come into the tubular cell, or you can simply generate it here by generating carbonic acid and having it dissociate. The bicarbonate is going to be put into the bloodstream, and the hydrogen ion is going to be treated in exchange for sodium. We've seen all of this before. Now, if the hydrogen ion and by the way, when the bicarbonate ion goes into your bloodstream, it can combine with the hydrogen ion and you can remove the CO2. It's a buffer. So this is just that molecule in your bloodstream acting like a buffer. It does it, okay? And then when it actually acts as a buffer, you can remove the CO2 with the respiratory system and lock that hydrogen ion up. If it just forms carbonic acid that stays in your bloodstream, you haven't removed it from your bloodstream. You have to actually have it dissociate and remove the CO2 to remove it from your body. Okay, this hydrogen ion that was either brought in from the extracellular fluid or put in here from this process, it's now in the urine, and it's going to be secreted. When you measured the pH of your urine, it was 6 or so, 6 to 6.5. Most people's urine is that much. Why is it so acidic? Because you're dumping hydrogen ions into it all day long. Everybody understand that? Now, this pump is active transport. Why is it active transport? Because we're using sodium go in, and you're going to have to use that energy going down its concentration to move hydrogen ion up its concentration gradient, because there's already hydrogen ions in there. And you gotta pump the sodium out. I know they're showing it going out now. Symport, that would be at the beginning of the tubule. Once the hydrogen ions build up in here, you gotta pump the sodium ions out. Okay? Now, if we did not have buffers in here, there's a limited amount of hydrogen ions you can put in because we're pumping the hydrogen ions up a concentration gradient. There's only so much energy in the sodium. And it can get it down to what, 6.5 or something? That's as low as it can go, I believe. Past that, there's no attempt to put it in. So how does it buffer? I'm glad you asked. So what we're gonna do now, watch what happens. We're going to put the hydrogen ion in, and there is sodium bicarbonate, there's bicarbonate ion in the plasma that form the filtrate. So then that bicarbonate is gonna join with the hydrogen ion. This is in the filtrate, forming carbonic acid, which forms water and CO2. The CO2 can be used to then produce another bicarbonate ion and put another hydrogen ion in. But what did we do to this hydrogen ion right now? Is it free in the filtrate? No, where is it? In the water molecule. So the carbonic acid bicarbonate ion buffer that is in the plasma went into the filtrate, and we're using it to have it absorb more hydrogen ions, and the filtrate, as in soon to be urine, can carry more hydrogen ions because we're forming or locking up those hydrogen ions in the water molecule. And notice, this bicarbonate ion, watch the sleight of hand for the bicarbonate ion. We now see the hydrogen ion get locked up in water. Where does the bicarbonate ion go to? We have taken a bicarbonate ion in the filtrate and put it back into the bloodstream. So while we took this hydrogen ion, created from water, put it in here so we could excrete a hydrogen ion, we took a bicarbonate in the filtrate and put it back into the bloodstream. Everybody see that? And what is this going to be used for?
Respiration.
Buffer your extracellular fluid. So we're not losing bicarbonate ions, are we? We recover them very efficiently as we are excreting hydrogen ions. Because then CO2 can go in and be used to create a bicarbonate ion in there. Well, that's not the only buffer there. Remember I said the phosphate buffer is there? So we're gonna have this hydrogen phosphate absorb the hydrogen ion, forming dihydrogen phosphate. That stays in the filtrate, okay? This is a weak base that absorbs the hydrogen ion. This stays in the filtrate and is excreted. And we can also have ammonia put in there. Now, these cells make ammonia. They take a glutamic acid and they deaminate it, NH2, put it in here, it absorbs a hydrogen ion, NH3, becomes ammonia, NH3, absorb a hydrogen ion, and form the ammonium ion in the filtrate. These cells are deaminating one non-essential amino acid, glutamate, and they then put that remaining molecule back in the bloodstream and the liver takes care of it, I'm pretty sure. Okay? But this is in the blood, in the filtrate, put in there by the tubule cells. This was in there from the blood. And then we're now using the bicarbonate ions that were naturally in the filtrate from the plasma because they go through the filtration membrane. We're using them here. So we have 3 buffer systems working on helping the urine carry more hydrogen ions. So we get rid of hydrogen ions bound up to these 2 or in the water molecule as we made a bicarbonate ion to replenish the, because we're not actively absorbing the hydrogen ions, are the bicarbonate ions. We're not actively absorbing them, are we? We do a little bit in the proximal convoluted tubule, but we absorb them this way, by putting them back into the bloodstream. Without these buffers, you could not get rid of sufficient quantities of hydrogen ions in your extracellular fluid. The buffers are absolutely required.
In which part of the tuber it happens?
This is happening mostly in the distal convoluted tubule. In the proximal convoluted tubule, we have this going on, but it doesn't excrete as many hydrogen ions as can the distal convoluted tubule. Yes?
Does the sodium leave the body with the urine in the bloodstream?
Yeah, we get rid of, well, there's still gonna be sodium ions absorbed. I mean, the distal convoluted tubule is still, if necessary, can absorb those hydrogen, those sodium ions.
Even with the phosphate?
But we have to lose sodium ions every day because we're taking in a lot of sodium ions. They just put those in there to make the molecules. Okay? Maybe we have to balance the charge so they have that. Okay, now let's talk about, the renal mechanisms. The renal mechanisms can remove huge amounts of hydrogen ions every day. But if you suddenly start generating too many, it takes a while, several days, before it becomes maximally active. If you need to excrete more, because you've got to get these tubular cells to have a large enough number of these channels, okay? So, renal mechanisms. Huge amounts of hydrogen ions can be removed. If you suddenly are generating much more, it takes a couple days for them to come and to be fully active. They're active enough to handle the average daily load you generate. So, what's the first thing that happens to a hydrogen ion when it's generated, goes into your bloodstream? A buffer takes care of it. If it builds up too much and the buffers are beginning to get titrated and there's less of them, then you're gonna start breathing faster because the pH will drop and you'll be removing carbon dioxide. And the kidney, once the pH drops a little bit in the blood, the kidneys will start becoming more active. Okay, and this is a giant chart of most everything. Okay, so if your pH goes up, your respiratory system is going to go down, reduce its activity. That's pH going up, so it's becoming basic, and buffers will respond, releasing more hydrogen ions. The lungs will remove some CO2, and you'll actually you'll be secreting fewer hydrogen ions. That'll allow the pH to come back down. If pH goes down, which is the normal case, then the buffers are gonna absorb those hydrogen ions. Your lungs are gonna breathe faster and deeper and remove some carbon dioxide and lock up on some excess hydrogen ions in the water molecule, and your kidneys will start removing more hydrogen ions, and it will come back up. Okay, so all three of them work together. Now, let's talk about disorders of acid-base balance. If your pH drops below 7.35, it's called acidosis. Okay, just going from 7.4 to 7.35. Doesn't seem like much of a change, but this is serious and can be life-threatening if the pH drops. Going above 7.45 is called alkalosis. Your blood is acidic or alkaline. That's the name of those terms. They both affect the central nervous system. They're serious disorders. Okay? Alkalosis and acidosis have a variety of causes. And remember, metabolism can generate a variety of acids. Buffers resist the pH changes initially, but only the respiratory system or the kidneys can remove the hydrogen ion from your body. Okay? Remember those principles. So let's look at the variety of acidosis and alkalosis. Respiratory acidosis occurs because your respiratory system cannot remove enough carbon dioxide and the carbonic acid, CO2 builds up in your blood, carbonic acid builds up, hydrogen ions build up in your blood if you cannot remove enough CO2. Okay? So if you have CO2 build up in your blood because your respiratory system is compromised for whatever reason, you're going to have respiratory acidosis, and quickly your blood pH is going to drop below 7.35. So if you have emphysema, not enough ability to get rid of CO2, your blood pH will drop. If you hold your breath for a long time, your pH in your blood will drop as CO2 builds up. It will happen every time. Okay? Your kidneys can compensate if it occurs gradually. If it occurs quickly, your kidneys can't suddenly ramp themselves up and get rid of a lot of hydrogen ions. Just can't do that. Okay? If you're drowning, CO2 will build up. Lungs can't get rid of it. Kidneys can't respond quick enough because it's building up very quickly. Okay? Respiratory acidosis. It's serious when it occurs. Now, respiratory alkalosis is also possible. In that case, you're hyperventilating and lowering your blood CO2 levels below normal. This is uncommon, and it's of no consequence because eventually respiration will be reduced and your blood pH will drop back down to normal range. So we have blood pH too high for a respiratory reason. That's what respiratory alkalosis is. So if someone is hyperventilating, yes, they have alkalosis, but eventually their breathing rate will go back to normal. Whatever causes it will be removed. They reduce their respiration, and the blood pH will go back to normal. It's of no consequence. Okay? It really is rarely serious. Okay, metabolic acidosis can occur. So this is blood pH dropping for a metabolic reason. What are you doing? For some reason, you're producing excess acids. Okay? And now, that occurs by, for some reason, you're producing excess acids or impaired ability to excrete ions, hydrogen ions, by the kidneys. Or a significant loss of bicarbonate from the extracellular fluid. In fact, this is the most common reason for metabolic acidosis is persistent, prolonged diarrhea. Because diarrhea has, your large intestine does secrete bicarbonate ions to deal with the acids generated by the bacteria. That happens. So you're secreting a lot of bicarbonate, and normally you reabsorb some of them, but if you have persistent diarrhea, you're going to be pulling bicarbonate ions out of your blood, and you're going to have a metabolic reason for causing a low pH in your bloodstream, extracellular fluid. Okay? And if you have a lot of anaerobic metabolism going, what acid are you going to be generating? Lactic acid. The most common cause of metabolic acidosis is that persistent diarrhea. It's not just having trouble to absorb materials or whatever. It's metabolic acidosis. Their pH drops. So a person that has persistent diarrhea, if they have metabolic acidosis, how can you tell from their breathing? Respiratory function is normal as far as their respiratory system. Their pH is going to be low, acidosis, they're going to have a high breathing rate. So a person who has persistent diarrhea and they're breathing faster than normal, they have metabolic acidosis. The reason they're breathing faster is because they have metabolic acidosis and the body's response. Kidneys are going to try and get rid of those hydrogen ions, but your breathing rate's going to respond quicker and it'll go elevated to remove CO2. When that happens, when you have metabolic acidosis and you're breathing faster, what happens to the buffering capacity of your blood? Because you're breathing faster. It decreases. Because you're removing extra CO2 to bring the pH back up. You're getting rid of the bicarbonate ions, so you have less buffering capacity in your bloodstream. These are interrelated. So, persistent diarrhea can lead to metabolic acidosis and compromise the buffering capacity of your blood and interstitial fluid. Okay? If it's too severe, respiratory compensation can help bring the pH near, but the kidneys have to sit and excrete those ions. You have pulled out a bunch of bicarbonate ions out of the bloodstream, pH has dropped, you've got to get rid of those extra hydrogen ions that is causing the metabolic acidosis.
Can you give someone bicarbonate? Does that help?
You could. That will help do what?
Buffer.
Raise the pH and also to get rid of some hydrogen ions, because you can now, once those bicarbonate ions hit their bloodstream, they can then remove the hydrogen ions by respiratory system. Once the bicarbonate is gone from the body, you have trouble getting rid of it. If you give them bicarbonate, yes, that would help them. Okay? Could you give them bicarbonate ions intravenously? Yeah, that would be even faster than giving it to their digestives. Okay? The treatment of those is beyond where we want to go. But just so you understand what is the common cause and what are the symptoms of someone who's in metabolic acidosis. Okay, what's the last one? Metabolic alkalosis. It's uncommon. Okay? It's caused by a loss of acid from your body. How do you do that? Repeated vomiting. So you eat, begin to digest, secrete the hydrochloric acid, vomit that acid out. What are you left with in your bloodstream? What did you put in there when you made the acid? Bicarbonate ion. So then you eat again, create some more hydrochloric acid, throw some more bicarbonate in your bloodstream, and vomit up the acid. You're building up bicarbonate ions in your bloodstream. Okay? That's the most common. Repeated vomiting is the most common cause for metabolic alkalosis. You could take too many Rolaids and also generate that, but it's much more common to simply have the repeated vomiting. So, if you have metabolic alkalosis, what will be your respiration be like?
Low.
It would be lower than normal because your pH has gone up. Hypoventilation can bring the pH to near normal. Okay, you can cut your breathing down, still get enough oxygen onto those well-designed hemoglobin molecules, right? And you can bring the pH to near normal as the kidneys reduce the hydrogen ion secretion they normally do. The kidneys will respond by getting rid of fewer hydrogen ions, retain some, and your respiratory system is going to allow the carbon dioxide to build up higher than normal. You're doing hypoventilation, allows CO2 to build up to generate more carbonic acid to solve the high pH problem. So get rid of fewer hydrogen ions and then put some more hydrogen ions in your bloodstream by letting CO2 go to higher levels than they should be. Everybody follow that? Okay, we are at the end of, there's only 4 kinds of alterations to acid base. Please know the common causes, how the body responds to it. That's the major things we covered. Major cause, variety of causes, what the pH of the blood really is, high or low, whether it's a metabolic problem, or whether it's caused by a respiratory problem, and kind of what the respiratory system does in those cases. So, let's go on to a couple of questions. Finish chapter 27 and begin 17. We have 4 chapters left to cover, 17, 18, 28, and part of 29. That's all we're gonna cover for the rest of the course. We're getting near the end. Anyone excited yet? Yeah, okay, we all are, I agree. Okay, what effect would the decrease in the pH of the body fluids have on the respiratory rate? It would go the opposite direction, is increase. You all got that, very good. Well, we did it in the lab. Okay. How would a prolonged fast affect the body's pH? Why would it get lower? What would lower the pH if you have a prolonged fast? Yeah, it's gonna produce hydrogen ions. Where from? When you switch from glucose metabolism, you move to, huh? Lipid metabolism, and lipid metabolism releases fatty acids. And then a little later, you're gonna start producing from the liver, you're gonna produce ketone bodies. Some of them are acidic. Okay? So that's the source of your body's pH dropping as you have ketone bodies and the fatty acids lower the pH. And what's that gonna do to your respiration? It's gonna increase it faster than normal. Okay. A child was revived after 5 minutes underwater. Yay, we got him revived. What acid-base disorder do you suspect the child has developed? Respiratory acidosis. Anything else?
Isn't metabolic,
Would they have metabolic acidosis? Yes or no? Let's say the child struggled underwater. What acid would you generate then?
lactic acid.
Lactic acid. That would have. So this child would suffer, assuming they struggled underwater, both respiratory acidosis and metabolic acidosis from the lactic acid. Wow. Big challenge. Yeah. And that acidosis can cause serious problems during that time. If it's cold water, you've heard of children being revived after a few minutes. It's a real challenge because of the acidosis that's in their body. Okay? So they have respiratory acidosis and metabolic acidosis from the lactic acid. I believe that's the last question. So, not this coming Tuesday, but the Tuesday after that. Give you a little time to study. Okay? Chapter 26 and 27, 40 questions each. Any questions on that? You ready to go into chapter 17? Okay, 17 is going to be dealing with the details, of the hormone endocrine system, okay? So the sheets are coming around. Well, I changed the PowerPoint, and we're going to start for the next 20 minutes. We're going to start and begin looking at the endocrine system. Now, this is into 2 chapters. Chapter 18, 17 is a reasonably, it's an average chapter as far as the length goes. 18 is a short chapter because how many of the hormones have we already talked about? A lot. The reproductive hormones, growth hormone, we talked about a lot of hormones already. So we have, most of it I'm gonna skip over. I don't have to describe aldosterone. I don't have to describe angiotensin. There's a lot of hormones I don't have to talk about because we've already beat them to death. So 18 will be a chapter where we just pick and choose hormones we have not described. And actually, one of them we're gonna deal with superficially, testosterone, estrogen, progesterone, because we're gonna get them in chapter 28. Okay, functional organization of the endocrine system. And here are the learning objectives. Describe the 4 classes of chemical messengers. I'm going to reduce it to 3. The book says, oh, you could take neurotransmitters and place them here, and I'm gonna do that. So there's really just 3 classes we wanna look at. Define a hormone, define a target tissue, distinguish between endocrine and exocrine glands. You know that already. Exocrine glands have ducts. They secrete a solution into a duct, goes onto an epithelial surface. Endocrine glands secrete it into the interstitial fluid, goes in the bloodstream, and circulates throughout your body. Compare and contrast the nervous system to the endocrine system. Those are both control systems. They have their own characteristics. They have some similarities, and they have some differences. Which one, for example, acts fastest?
The nervous system.
Nervous system. Which ones acts longer in time? Endocrine system. So there are differences between these as well as similarities. We'll just kind of touch on that. Describe the common characteristics of all hormones. There's 3 of them. Define a binding protein, a bound hormone, a free hormone, and discuss the effects of binding proteins on circulating hormone levels. Probably won't get that far today. Listen and describe the 2 chemical categories of hormones. And they are lipid soluble, water soluble. They have radically different characteristics. Explain the influence of the chemical nature of a hormone on its transport in the blood. Do lipids get transported easily in the blood? No. Do water-soluble hormones? Yeah, they go. So they're transported in the blood differently. Removal from circulation, water-soluble are removed quicker than lipid-soluble. And its half-life, how long does it take after you put a certain quantity in? How long does it take for half of it to be removed? We'll talk about that in general terms. Discuss the 3 main patterns of hormone secretion. List and describe the 3 stimulatory influences on hormone secretion. Give an example of each. What are the 3 ways hormones can be stimulated, be secreted? And what are the 3 ways that hormones can be inhibited from being secreted? They're exactly the same 3. Okay. Discuss the major mechanisms that maintain blood hormone levels. Describe the general properties of a receptor and how a target cell may decrease or increase its sensitivity to a hormone. Or a cell can decrease or raise its sensitivity to a hormone. And I'll describe that one. Explain the mechanism of action for the 2 types of receptor classes, lipid soluble, water soluble. The water soluble has a lot of ways it can produce an effect. Define amplification, this occurs only for water soluble, and explain how despite small hormone concentrations, water soluble hormones can cause rapid responses. So let's begin looking at chemical communication. Cells have to communicate with neighboring cells, and there are 3 methods or types of molecules for communication, okay? Your body, in an attempt to communicate with neighboring cells, have 3 ways of having chemicals communicate with neighboring cells. Autocrine messengers are where you have a cell release a chemical that affects its physiology, okay? And the only one we've seen of that is a T4 cell, after it's activated, can release interleukin-2 to stimulate its own division. It's self-stimulating. That's an autocrine messenger. There's not too many of those examples in the human body, okay? Paracrine messengers, one cell releases it, the chemical, it goes through the interstitial fluid to get to a target cell, and it affects it. We have actually talked about one of those, and that would be the histamine released by the enteroendocrine cells that affect the parietal cells in the release of gastric juice. Okay? So the histamine is a paracrine factor. Neurotransmitters are also chemicals released by one cell that affect the nearby cell. So I include, and most everybody else includes, neurotransmitters as paracrine factors or messenger. Okay? The book calls them separate, but they fit exactly the definition of a paracrine messenger. They're released by one cell that affects a nearby cell. So we're just gonna call them paracrine, and there's just two classes. Yeah, they're released by neurons, but are all neurotransmitters released by neurons? No. There are receptor cells that release neurotransmitters. What do you call it then? Is it a neurotransmitter or a paracrine messenger? It's a paracrine messenger. Okay. Endocrine messengers are chemicals released by one cell. It gets to its target cell by going through the bloodstream. It can't go through interstitial fluid to get there. It has to go through the bloodstream. And when we had gastrin released by the duodenum, or secretin released by the duodenum, it had to go through the bloodstream to get to the stomach. It had to go through the bloodstream. Okay? Histamine didn't, because histamine is a paracrine messenger, but gastrin, secretin, cholecystokinin, and all that stuff we saw was an actual hormone. Okay? These are hormones, paracrine factors, those are autocrine factors. Okay. Remember that endocrine cells release a chemical into the interstitial fluid. It goes to the bloodstream, circulates throughout your entire body. Excuse me, dry throat already. It leaves the bloodstream at every tissue. And if the cell is a target cell, it responds to the hormone. The target cells produce a response to that hormone. What happens to cells that are not a target cell for that hormone? They don't do anything. What makes a target cell, do you think?
Receptors.
A receptor for the hormone. So, you have a hormone released into your bloodstream. It circulates and leaves every capillary bed and is exposed to every cell in your body. Only the ones that have a receptor respond. Okay, parathyroid hormone circulates throughout your body. Only certain cells respond to it. Only ones that have it. So you can have a hormone released that affects two widely different separated tissues, because both of those have receptor cells and all the other cells don't. Excuse me. Only 5 minutes more to go here. Okay. This is a table that summarizes autocrine factors, paracrine factors, neurotransmitters, which should be up there, they really are, and endocrine cells. Okay? Let's compare the nervous system and the endocrine system. They're both control systems. They have some similar characteristics, but definitely a lot of different characteristics also. Nervous system is going to release a neurotransmitter at nearby effectors. Now, the nervous system using neurotransmitters, it is a chemical. It's a paracrine factor as far as I'm concerned. The endocrine system releases hormones that affect distant cells. By nearby, it's near the end of the neuron, the axon. Both systems use brain structures. Did we talk about any hormones in chapter 27 released by the brain? Or 26? ADH. produced in the hypothalamus, shipped down the axons to the posterior pituitary, and released. Okay? They use similar chemicals. Norepinephrine is a neurotransmitter, and it's also a hormone released by the adrenal medulla. Some neurons release hormones. We just talked about ADH. Does the anterior pituitary release any other hormone? Oxytocin. Oxytocin, and that's a hormone. And both of them, both systems, can use G proteins in producing the change inside the cell. Now, the two different systems use different means of transportation. The neurons, nervous system, uses an axon to get it to point to where it needs to go. And the endocrine system uses the bloodstream. They have different speeds of response. Neurons, nervous system, very quick changes. It takes, for even the fastest hormones, it takes nearly a minute to days to get the effects, okay? And they persist for different lengths of time. If you want to keep having a neurotransmitter have an effect, you got to continue to send action potentials. Hormones released until it's removed from the blood, it's going to produce a response. Last slide, guarantee it. Hormones are amplitude modulated, which means if you want a bigger effect, release more hormone. Every time you release more hormone, you get a bigger effect, okay? For the nervous system, it's frequency modulated. If you want to get more effect, have a faster stream of impulse, more neurotransmitter release. So those are the two differences between those two systems. We're going to begin with the characteristics of hormones next time. If you have not seen exam